Welcome everybody. We are on lesson 9 of our muscular system. This lesson we have returned to the muscle, and we're going to get in deep with how exactly that muscle is contracting. To understand this, we need to talk about what is known as the sliding filament model. And we're going to talk about those filaments that we're going to talk about are actin and myosin that we've already discussed. We know that those are the two main components, and we're going to talk about how they actually work together to make the muscle contract. So when an action potential reaches a muscle, and again, that's through a motor neuron, it's going all down the axon, we go through the neuromuscular junction, and when we get across that neuromuscular junction, we are actually sending a action potential across the sarcolemma, which the sarcolemma is another name for the membrane that surrounds the muscle cell. So those sarcomeres are the individual units within those muscle cells that are actually going to contract. And so we're going to make the actin and myosin work the way that they're supposed to so that they actually contract. So here is a little video that I'm going to show you that I hope is not, no, I think I turned down the volume here. So what we have here is this green filament here is the actin, okay? And this other one is the myosin, and they are in very close proximity to one another. The myosin head is the most important part because that is where the interaction between the actin and myosin actually occurs. And there is actually these binding sites, and that's these specially colored actin molecules. Myosin can bind at these sites. So when myosin actually receives the signal to contract, you can see it makes a little motion. It just pivots a tiny bit. That molecule itself actually changes shape. And this is all powered by ATP there. You can see that. You need the energy there. And when ATP is used, it actually then creates another link and can repeat the process so that another ATP molecule will need to be brought in, and it can continually link up and drag the actin in one direction. So this is actually, it's sort of a pulling motion. And we're going to see how that works together. So this is the sarcomere, and this is a summary of all of this together. So if you start up here at the top, we see a relaxed sarcomere. So you can see that we have in this case, the actin is purple, and then the myosin is green. And you see that the actin is on one side, and then it's also on the other side, and there's space in between. And that space is filled by the myosin. So what is going to happen is that we are going to see that the myosin is actually going to pull the actin, and it's going to pull it towards the middle of the sarcomere. So these actins right there are going to move to the left. These actin are going to move to the right, and they're going to meet in the middle. And you can see that's what's going on throughout these diagrams, is that we see that the myosin is pulling in this direction, pulling in that direction, pulling the two sides together. And what you notice is that the two sides of the sarcomere keep getting closer and closer together until at the bottom here, we have the sarcomere that is fully contracted. And this is happening over and over in repeating units throughout the muscle. So even though an individual sarcomere is only shortening by less than a millimeter, that distance, when you multiply it by tens of thousands of times, is enough that it actually creates a very forceful shortening of the muscle. So actin and myosin are roughly the same length, and they don't actually change length. That's like a big misconception out there. The actual fibers themselves don't, but the amount of overlap does change. So if you notice that when we are relaxed, the amount of overlap between actin and myosin is at its minimum, okay? And this is a relaxed muscle. You can actually even stretch out the muscle. When you have a muscle fully stretched, you're actually going to stretch this to the limit where these myosin are only going to be right on the edge of the actin, okay? And then what we see is that when we contract, we're actually going to increase the amount of overlap between the two fibers. So the way we describe it here, the actin myofilaments, and we use these terms interchangeably just so you know that an actin filament or an actin myofilament, it's the same thing. The most technical name is a myofilament, but if you hear filament, don't get confused. So basically, what's going to happen is the actin on one side and the actin on the other slide past the myosin and move towards the middle, otherwise known as the M line. And then the Z discs, which are the ends for each sarcomere, are going to move close together, and the whole thing shortens. Okay? So as this happens, what we see, and this is where we first figured this out, because although we can't see actin and myosin individually through the in a microscope, what we could see is these light and dark regions. And remember that the myosin is thick, so it's darker. The actin is thinner, so it's lighter. And of course, the darkest is when you have actin and myosin together. And when you look carefully under a microscope, you can actually distinguish these different shades, the light and dark. And this is what's called, that banding, the striated nature of these muscles. So the H zones are these zones right here. The H zone is myosin only. And what we see is the H zones narrow. And also the I bands. The I bands are on either side of a Z disc, and they are actin only. I bands are actually the lightest because actin is, as it's sometimes called, the thin filament. So these are bright under a microscope, these I bands, and these H zones are darker than the I bands because they've got myosin, but lighter than this area where we have actin and myosin together. So that's where we start to see this. And the A bands they don't actually change. They never actually change the and so what we see is that the A bands are equal to the length of the myosin, but they don't actually narrow. So these are actually are essentially staying the same size. So in a fully contracted muscle, ends of the actin myofilaments actually overlap in the H zone, so you actually see a little bit of overlap there. And the H zone is essentially gone. There is no H zone anymore. So that is a fully contracted muscle. And as you can imagine, that's the limit of the muscle. There is no more ability. Notice that these myosin can't pull anymore because they've already reached the Z discs. So this sarcomere is fully contracted and cannot contract anymore. So one of the things that we sometimes talk about is that muscles are strongest in the middle of their contraction, where there is the most amount of overlap in the actin and myosin. Because when we get to this point where we're getting to the end of that muscle contraction, there's no more ability to forcefully pull here, so the muscle starts to lose strength. And if you've ever lifted weights, you know this, that when you're doing, a bench press, the hardest part is that beginning and that very end. It's not in the middle. The middle is usually the easiest. So, and a good example is if you ever analyze the starting position of a sprinter, look at where their muscles are. Every one of their joints, where their major leg muscles, their major running muscles is actually at a 45-degree angle. So it's basically trying to set up a situation where when you get off the starting gun, you actually maximize your strength there. So here's another video here, and it's just going to show all of this together. And again, I've turned the sound down. So here we have a relaxed sarcomere. We got the actin, the myosin. Okay, and what we're going to see is that H zone is wide open now, the I band is larger, and we're going to start to see H zone disappear, I band disappear, and then these Z discs coming closer together. And you can see how it's all working together, and when you repeat this over tens of thousands of sarcomeres, that you have that. But again, you reach that limit, and that H zone disappears, and the I band doesn't disappear, but it greatly narrows. So there's another video that we're going to put as a separate video, but that's the end today. So thanks, guys. I'll see ya.